Okay, so hello, good afternoon, everybody. Welcome to the last session for this course. As you're already familiar with me, I'm Ankita. I have been your TA for this course for around 12 weeks, 13th week together. So, first of all, let me thank all the students who had joined the course, had joined the live interaction sessions, or were able to access the notes online once the slides were uploaded, or were able to access it via YouTube. So I hope it was helpful to you in some manner or the other. Either with direct interaction or otherwise. It has also been an enriching experience for me, so I would like to thank everybody for that as well. Now, so today's idea, it's an extra session where the intention is so we'll basically be covering some of the leftover topics, particularly from the week that we discussed, hypersensitivity. So as I had discussed during those classes, we had a couple of topics left over, majorly the type 2 and type 3 forms of hypersensitivity. So that is what I'll start off the discussion with. And then we'll go through the overall, all of the topics that we had discussed during this entire course. And if there are any doubts and discussions that are necessary, we can take them up at any point of time. Please feel free to interrupt me if you have a question. But for starters, we'll just cover those two topics and wrap it up really quickly. And the rest of it we can come to after that. Okay, so just to quickly brush up what we had already discussed, these are the types of hypersensitivity reactions like we already are familiar with, type 1, 2, 3, and 4, whereby type 1, 2, and 3 are the subtypes which are elicited, which reactions are elicited immediately as an immediate response of the immune system, versus the type 4 is delayed type hypersensitivity, which sets in much later. Among these, we had gone into details about type 1 and type 4, particularly in the previous classes. So we'll mainly be focusing on type 2 and type 3 today. So type 2 is IgG mediated cytotoxic hypersensitivity, and type 3 is immune complex mediated hypersensitivity. So we'll be coming to that in the subsequent slides. You're already familiar with this table. It was part of one of my previous slides as well. So just to quickly brush up, this is what used to happen in hypersensitivity type 1 reaction or 1 response, whereby as part of the as part of the type 1 response, we used to see how there was an interaction of the allergen with the antibody, which was able to recognize it. And in response to that, we were, just a second, and as a response to that allergen being recognized by the immune system, we were able to see how mast cells were sensitized, and in its response, degranulation was stimulated, and in response to that, vasoactive amines were released, and as a result of that, we could see downstream reactions, which altogether comprehensively manifested the type 1 response. However, in contrast, when we talk about type 2, like I already mentioned, it is IgG mediated, cytotoxic in nature, and is an immediate form of response. What does it involve? It also involves antibodies, but the difference lies in the fact that it leads to cytotoxicity or cell death. So IgG mediated cell destruction is a major component that distinguishes type 1 and type 2 forms of hypersensitive reactions. Under that, two mechanisms majorly come in. One is ADCC or antibody dependent cell mediated cytotoxicity, and the complement mediated activation. So you must be familiar with the complement system as well by now. So these are the two ways by which cytotoxicity is elicited as a response to the type 2 hypersensitivity reaction. Now, these are common examples for that. The first example of type 2 hypersensitivity response is blood transfusion. So you must already be quite familiar with it, that people under certain conditions when they're suffering from a disease or are undergoing surgery or any sort of trauma has been faced as a result of some accident or something of that sort, blood transfusion is often carried out and is quite extensively carried out. And majorly when we talk about blood transfusion in context of hypersensitivity, it is with respect to the ABO blood antigen typing. So in case you're not familiar with it, you know there are four blood types, A, B, AB, as well as O. And under each of those subtypes, you have an Rh positive and an Rh negative subtype. And based on that, each of the blood types are classified. What exactly happens under that is if there is some sort of a cross reaction which occurs, that is if you're already familiar with it, the way in which these antigens work is that if there is some sort of incompatibility, that is a patient who possesses an antigen which can elicit a response against an unfamiliar antigen. In such cases, what happens is there is agglutination, or in other words, the clumping of antibodies within their system, which eventually leads to cell lysis, and that has the potential to become a very lethal condition for such patients, and therefore blood type matching. HLA type matching is more relevant when we talk about organ donation, but when we talk about blood donation, blood type matching is very, very important and is extensively being carried out. Of course, under that, you have universal donors and universal acceptors as well. So these are concepts which we usually learn in preliminary classes, so I'm not getting into that. But yeah, in case there is any sort of mismatch, like I said, it leads to complement-mediated cell lysis. The second type of example under type 2 hypersensitivity is for drug-induced hemolytic anemia. As you can understand from the name itself, there is lysis, cell lysis, which is a major component of this disease. So while anemia by itself is quite a common condition, hemolytic anemia is very, very severe. And in some cases, it can be genetic, but this in particular is in response to a drug. Let's see what exactly happens. So in such cases, in such a patient, it will occur when an antibiotic is forming a complex with the RBC membrane or surface protein, much similar to hapten carrier complexes, which again is something we have looked at before. So that also leads to the formation of IgG antibodies because they are recognized as something foreign within the system, leading to complement-mediated lysis. Majorly, this is same for drugs like penicillin, cephalosporin, or streptomycin. So you might have come across people who you might have heard of having penicillin allergy, and therefore they cannot be, given this entire group of drugs, which makes it slightly more challenging to treat such patients. But this does happen because in that case, it results in a hypersensitive reaction, and therefore they are allergic to that type of drug. And such sensitivity should always be mentioned in case such a patient ever needs to be hospitalized. So these are two common examples of this sort of hypersensitivity. The example that I want to focus on taking it as the most important example in this context is something you might have studied before as well, which is erythroblastosis fetalis, which can be a very, very lethal condition. And while we are overall familiar with the concept, we'll go into the immune aspect of it today to understand it better. So I think before we go into the schematic, we'll go over the process once in the upcoming slides, and then we can come back to the schematic for better understanding. So hemolytic disease of the newborn, or erythroblastosis fetalis, is not something that sets in just in one go. It actually involves two separate gestational periods or two separate pregnancies within a woman. Now, what is the background context that is necessary for this condition to at all manifest is that if there is a father who is Rh positive and a mother who is Rh negative. So when such a couple, they conceive a baby who in turn is Rh positive in nature, there is potential for this sort of response to come in. So what exactly happens there is that the Rh positive fetus very naturally expresses an Rh antigen on its blood cells, which are absent in the mother. During the first pregnancy, fetal RBCs would be separated from the mother's circulation because of the placenta, and particularly within the placenta, there is one layer of cells which are called as the trophoblast. If you're not already familiar with it, it's fine. You don't need to know all this additional terminology. But basically, there is a mechanical barrier that separates the fetal blood from the maternal blood. Now, during this first pregnancy, it usually such a woman would not be exposed so as to allow the fetal RBCs to activate her Rh negative specific B cells because of this placental separation. However, when such a patient is at the time of delivery when they're in labor, there is potential that when there is separation of the placenta, especially if you're not already familiar with it. So whenever a child is delivered, after that there is a second round of delivery where the placenta, which also sort of serves as an organ, has to be delivered separately from the patient. So during that time, because the placenta is usually, think of this as the uterine wall, the placenta is attached to it because it allows the baby to easily access the mother's nutrients, and also for the placenta to accordingly adjust in size as the baby grows. However, when this separation needs to happen, at that time, the uterine wall detachment from the uterine wall can often allow a large amount of blood from the fetal umbilical cord to actually enter into the mother's circulation. So this is where it becomes dangerous during the time of delivery. Then, Then what can happen is that these fetal RBCs, which are Rh positive in nature, can activate the Rh negative B cells in the mother. Now, this results in the production of Rh specific or anti-Rh plasma cells and memory B cells, which will remain as a memory immune response within the mother. Now, this case by itself can end right there. The problem comes in during the second pregnancy. We'll come to that now. The secreted IgM antibodies, which are part of the mother's immune system, and IgM, if you're already not familiar with it, you would be. We have discussed this extensively. That IgM becomes the first antibody class which is expressed on an activated B cell, will help in clearance of whatever Rh positive fetal cells are present from the mother's circulation. But what remains as the threat are these memory cells. Why? Because class switching will occur from IgM, which is normally expressed, to IgG, which is capable of transplacental migration. Again, if you don't already remember it, we had discussed certain antibody subtypes which are able to move from one location to another. We had talked about secretory antibodies, we had talked about transplacental migration, etc. We'll again be brushing over it today itself if time permits. So that can act as a threat or a very huge problem for any subsequent pregnancy with an Rh positive fetus. So in case that does happen, that is, during a second pregnancy, if there is activation of these memory cells in this subsequent pregnancy, it can lead to the formation of these anti-Rh IgG antibodies. So these antibodies would then be able to cross the placenta and damage the Rh positive fetal RBCs. In some cases, it can be a very mild condition that they do not have sufficient RBCs. Anemic conditions can persist, such a baby might grow malnourished. But what eventually can happen is that it can lead to developmental anomalies because if you have studied about hemoglobin and the way it functions and the way that it acts as the major source of iron within your body, apart from ferritin, of course, you would understand that it can lead to developmental deficits in a lot of cases, and in a large number of cases, if the condition is very severe, it can be very fatal. So that is a major problem. And in addition, the conversion of hemoglobin to bilirubin can also present as an additional threat to the newborn, because this lipid-soluble bilirubin can accumulate in the brain and cause brain damage as well, like I said, developmental deficits. So taken together, if we sum up what exactly can happen in this reaction, we'll come back to the schematic for this. So this is the development of erythroblastosis fetalis. You look at the first pregnancy and you see this is an Rh positive fetus within an Rh negative mother, and you can see how there is a separation between this placenta and the maternal circulation. And within the baby, you see RBCs with Rh positive, which are Rh positive in nature. At the time of delivery, you can see that these RBCs have actually crossed over into the maternal circulation. As a result, what happens is that there is an immune response elicited against it, and it becomes Rh specific or anti-Rh B cells, which are being synthesized. Some of them would be plasma cells, which carry the IgM subtype, and they will remove any cells, any Rh positive cells from the fetus and clear it out from the system of the mother. However, whatever memory cells are present will actually remain, and these memory cells will persist as a potential threat for a second pregnancy. or subsequent pregnancies. So during the second pregnancy, what can happen is that because this mother circulation already carries some of these memory cells, it has potential of crossing over from the or carrying out transplacental migration. And as a result, these IgG molecules, why IgG? Because IgG is capable of transplacental migration. So these IgGs, which are anti-Rh in nature, can cross the placenta and attack fetal RBCs, leading to the condition known as erythroblastosis fetalis. So this is what the exact condition is. Now, there also has been a solution to this because very early on when the disease was initially studied, it was actually observed that subsequent pregnancies were not taken to completion and babies used to suffer from deficits or often used to die, and therefore a solution has come up for it. So you can see here that prevention or just a prophylactic method of avoiding this condition is to use RhoGAM. RhoGAM is it's an injection, basically sort of a preventive strategy whereby the mother is treated with RhoGAM, which is sort of an antibody mix basically. So what it effectively does is that it prevents the activation of these B cells, which have been synthesized as a response to this to the Rh positive cells within the mother circulation, and therefore memory cell production will be stopped, and therefore there will be no potential threat for the subsequent pregnancies. However, what you need to also keep in mind is that the time of administration is extremely crucial. It has to happen within 1 or a maximum of 2 days, because beyond that, as you can expect, as we have already studied the timeline of immune response previously, that after a period of 2 days, a response will be elicited, and then it will of course be too late because it will be difficult to remove memory cells as well from circulation. Also, what you need to keep in mind is that if there are subsequent pregnancies in such a mother, then in each case, if the fetus happens to be Rh positive, then in each case after delivery, RhoGAM administration has to be carried out to prevent any sort of danger in the future. So basically, this was the example of type 2 hypersensitivity response. So the prevention strategy in a little more detail. So almost entirely, this condition can be prevented by administering antibodies, which are IgG antibodies against the Rh antigen within the mother within a time period of 24 to maximum 48 hours after the first delivery with the Rh positive fetus. And this timeline is important because it has to be done before B cell activation is actually successful. These antibodies, like I said, are called RhoGAM, and what they effectively do is that they bind to any fetal RBCs that enter into the mother's circulation at the time of delivery and allow their clearance to actually be facilitated. No RhoGAM treatment, however, would be necessary if this baby is Rh negative. Then in such a case, you do not expect any response to be elicited, and that pregnancy would be carried out, without without much problem. So that's why mothers who are Rh negative and have partners who are Rh positive, in such cases, pregnancies or their entire gestation period actually is taken it is actually handled with a lot more care because it can be quite dangerous in certain cases. However, with correct prevention, such patients, they undergo complete treatment in a very proper manner. It's never a problem. So that was about type 2. So what we basically saw is that it is IgG mediated. It involves cytotoxicity. The response is immediate in nature. It is not a delayed reaction. And the cell death can occur either via ADCC or via complement activation. We saw examples for both of those. In terms of either it is blood transfusion related or it is related to hemolytic anemia, which is induced by some sort of a drug, or as we saw in the third case, erythroblastosis fetalis, which you'll find to be the most commonly cited example for this subtype of hypersensitivity reactions. If there's any doubt, please interrupt me. We can go over the topic again, but make sure it is clear in your head, because this is actually relevant not just for the course, but beyond that also, if you want to appear for some All India examination or something of that sort, you'll very often find a question on something like this. So please interrupt me if necessary. Now coming to the other subtopic, which was left over, which is the type 3 hypersensitivity reaction. So again, this is also an immediate form of hypersensitive response. The only difference in this case is that it is immune complex mediated. The difference being that the previous examples were just dependent on the antibody. Like, for example, the first one was dependent upon IgE for an allergic response. The second type, as we just saw, was dependent upon IgG. And this one depends upon the antigen antibody interaction or the entire immune complex. So basically, what is the idea? So whenever small quantities of antigen antibody complexes are deposited in tissues which are very, very close to the site of antigen entry, then a localized reaction is developed against that antigen. When large quantities of antigen antibody complexes are formed in blood, and because blood, as you obviously know, is part of a circulatory system or the circulatory system within the body, and it can potentially spread in a very systemic manner or throughout the system, then a very generalized reaction can occur, which leads to the deposition of complexes in certain, preferentially in certain locations within the body. What are these locations? The first is on the blood vessel walls. I think we had discussed it a few classes ago, last class, I think, where we had talked about how there is deposition of cholesterol along the walls of the blood vessels, which leads to conditions like atherosclerosis or, basically conditions whereby eventually the blood pressure is suffers and that patient's capillaries can get blocked, they can have other sorts of blockage in their heart, etc. So all those conditions are they're quite prevalent, especially if you have a tendency to consume fats which do not get digested from the body eventually, example being trans fats. So in such individuals, the conditions are a lot more common. However, in this case, we are talking about the deposition and the circulation of immune complexes, antigen antibody complexes, particularly either on the blood vessel walls, which is the endothelium, in the synovial membrane of joints. This is the synovial membrane. Basically, you can see this is somewhere around the patella, which is at the knee. And the third example is on the glomerular basement membrane of the kidney. So I think you would already be familiar with it, but if not, you can read up on it a little bit, that the glomerular filtration unit actually is the way in which you can see that the entire all the water that you consume within the body is actually undergoing processing within your kidney. Toxins are getting filtered out. As much of the nutrients are possible, they are being reabsorbed into your body with the idea that you want to minimize nutrient wastage as much as possible. And water loss should be also minimized so that your urine is concentrated, carries as many toxins as possible, and all the good stuff, all the necessary nutrients are being retained within the system. So all of that takes place within the filtration unit, which exists in all our kidneys. So that's basically their job. So these are the locations where we can often see that such conditions, such hypersensitive conditions can often set in. So yeah. So the deposition of immune complexes can actually initiate, I think there's some issue here, can often initiate a complement reaction. So there are examples of such conditions. So you have two sets of reactions. In the first set, you see there's C3A, C4A, C5A. Again, if you're not already familiar with it, we had looked at the entire cascade of complement system which persists, and we had seen that in each step, each of these proteases or enzymes were actually cleaving the protein which is right downstream of it and was generating two unequal parts, 1A and 1B. So it depending upon which stage we are talking about, one of them can act as a physiological mediator, which is often a part of the which is often helping convey information throughout the system. And the other part often becomes part of the next enzyme or next protease, which helps in cleavage of the subsequent step, therefore making it an entire cascade or an entire flow chart, which consists of multiple steps which are dependent upon the previous one. So examples of such complement proteins include a first set of reactions whereby you can see C3A, C4, and C5A are the smaller complement split products, and they are serving as anaphylotoxins, which means they cause localized mast cell degranulation and the subsequent or consequent increase in local vascular permeability. These are actually type 1 hypersensitive reaction characteristics, basically. There is also a second set of these things. Second set of reactions, which involves C3A, C5A, C5b, 6, 7, which act as chemotactic factors for neutrophils. What it effectively helps in doing is that it allows or results in the recruitment of neutrophils at this particular site. The tissue there becomes injured because neutrophil recruitment is followed by injury, tissue injury, because there is extensive release of lytic enzymes from these recruited neutrophils, and that entire area becomes injured, and therefore is a region where there can be deposition of immune complexes. Now, Now, this you see is an example of a localized type 3 reaction. The example taken here is of an insect bite. So after there has been an insect bite, a sensitive individual um, can often, suffer. They can have different types of reactions. In particular, we are here talking about a localized type 1 reaction, which usually, while it sets in immediately, it also is easier to recover, and therefore usually it recovers within an hour or so. However, in some cases, some 4 to 8 hours later, a typical Arthus reaction, which is localized type 3 reaction, can develop at the site, causing quite extensive and quite pronounced erythema and edema. So again, if you don't remember the terms, it is basically swelling, redness, swelling, and heat generation that occurs at the site of inflammation. And this again can persist for another 24 hours or so. Other examples of this type 3 reaction, and in particular here we are talking about the generalized examples or examples of generalized type 3 reactions, and one prime example for the same is serum sickness. So what exactly happens there? Again, this might be a little unfamiliar for you, but let's just take it one at a time. So during serum sickness, what is the key difference between them? It's not a localized form of type 3 hypersensitivity reaction, but instead it is systemic in nature. So it is often observed after the administration of antitoxins, which is a part of passive immunization from a foreign serum. So basically, you can see its examples for, say, for example, when a particular individual, say somebody got bit by a snake. So in that case, as an immediate way of eliciting a response, in a lot of cases, antitoxins or you know, passively passively generated antibodies, either raised in, say, a horse or some other animal, that can often be administered into a particular patient. So typically what happens then is that after exposure to the foreign serum antigens, many a time some individuals are found to manifest a combination of symptoms that are altogether called as serum sickness. It is not exactly a disease, it's more like a condition, more like a group of diseases where you can see a set of distinguishing set of features. So the symptoms for this condition include fever, some weakness, generalized vasculitis, for which you can see images here on the right, and you can see how there's edema and erythema in such cases as well. And it also is often associated with lymphadenopathy, which involves swollen or enlarged lymph nodes, arthritis, deposition in the synovial joints, and in some cases glomerulonephritis, whereby severe kidney diseases, usually they affect both kidneys, are often found, and it is characterized by inflammation within the glomeruli. So this is an example of a condition known as serum sickness. Again, keep in mind that it's not a very singularly characterized disease, but more like a combination of symptoms that are studied. So that is an example of type 3. So with this, we come to an end for the topics that I wanted to discuss for as additional topics that were actually left out as part of the course. If you have any doubts, please interrupt me right now, and we can take it up before we quickly revise overall all topics that we have discussed as part of the course. And of course, you can do that in the as and when we discuss each of these topics as well.
Excuse me. Hello.
Yeah, hi.
Hi. My doubts are not related to immunology like as a subject. The
Mm-hmm.
exam is coming up, so I
Mm-hmm.
can I ask something about the exam? So the exam will be through like CBT mode, right?
I guess so. So actually, we are more concerned with the course topics. If you have very
Okay.
like examination conducting related questions, I think it's better to mail a team member. They'll be able to get back to you better. But yes,
Okay.
it is via CBT mode only. But yeah, you can ask your
Okay.
other questions. If I know them, I'll try to address them certainly.
I was just asking what will be the pattern and how can we like approach the questions and I don't know. Like I've just applied for LBT and this course for the first time, so I don't know.
Okay, so basically, again, like I said, it's always in CBT mode itself, and
Okay.
majorly they are all MCQ questions only. You will find most of the questions to have one correct answer. In some cases, it's not
Okay.
always the case. There are some multiple correct answer questions as well. It would probably be mentioned
Okay.
in that case. And there is a certain section where you will find slightly longer questions, like which you need to work out a little bit. Those are few in number.
Okay.
If I'm not wrong, I think there were 10 questions probably like that, where you have probably some application based question where you use a concept you've
Okay.
studied and you try to address it. And yeah, for preparation, certainly go through all the assignment questions. That should help you cover up, at least prepare properly. Apart from
Okay.
that, you can go through the sample questions that I have given in the all the slides that are already uploaded. You can go through that, and you can look up any previous year questions if you're able to access them. You can work out any like if you have those books for, say, for example, CSIR exams or GATE or any of those, you can use those as the reference for your MCQs. So that's good enough, and go over the
Okay.
slide videos and the topics. Make sure that you've studied the topics. So questions, I think, would
Yes.
most importantly be there. Yeah. And you can
So
refer
I
to
was
textbooks also. Like I have used Kubi in all my slides and basically during the teaching.
okay.
And if you go through Professor Shudeep's lectures, he has majorly referenced Janeway's immunology. So you can refer that. You'll find questions there as well. In their exercise sections, you'll find some questions. So that can also be helpful.
Okay. Thank you.
Okay. So if there are any more questions, again, you can interrupt me at any point of time. I'll quickly brush over this, all of the topics that we have covered so that, there's a quick revision and then we can conclude the session for today. So, very initially, we had started off with a discussion about the immune system. We understood all its different features, starting off with the fact that it has two major jobs. Video. Sorry. So it has two major jobs. One is to recognize and then is to respond. So with respect to recognition, we something that we have also focused on later on is that it has to be able to distinguish between self and non-self, and that of course the all the sub-steps that are involved as part of that recognition is something we have also studied. How each of our immune cells are sensitized to our own antigen, so that we make sure that we don't, under normal circumstances, do not have a incorrect or unwanted response within the body. And in case that does happen, it leads to an autoimmune condition, which is, of course, very dangerous and, of course, compromises the quality of life in such patients as well. We had discussed after non-self recognition, we had discussed how a response comes in, where it comes in two stages, starting off with an initial or a primary response when there is an initial exposure to an antigen, followed by a rapid and a heightened response, like you can see. Just a second. Yeah. a heightened response in the second case, which is a memory response, because the immune system retains a memory of the first infection and therefore can respond much quickly the next time around. Following this, these were the different discoveries of all the scientists who have been awarded Nobel Prize. However, this is not the most updated condition. This was taken from an older version of COVID as well. You can, of course, update yourself regarding this. And we had discussed some of these discoveries extensively eventually. For example, we looked at the discovery of vaccines. We looked at the discovery of the entire system of gene segment rearrangement that we have covered over weeks as part of the course, and the discovery by Susumoto Negawa, how it contributed to that, the structure of antibodies, etc. Following this, we understood the distinction between innate and adaptive immunity, how innate by birth is a defense mechanism that you are born with, versus adaptive immunity, as the name suggests, is brought about or is actually brought together in response to the kind of exposure to pathogens that you get over time. And the key distinctions between them, how this acts as the first line of defense, whereas adaptive immunity sets in much later. Innate immunity is, while very useful, it's not complete by itself. There has to be the involvement of adaptive immunity in a large number of cases, and it helps in making the innate immune response even more efficient as well. And we also understood what are the shortcomings of each subtype as well. Following this, we looked at the components of the immune system, how there are different, you can think of them as sentinels, which are all together comprehensively keeping the immune system together. So we started off with the anatomic barriers, physiological barriers, phagocytic and endocytic barriers, and inflammatory barriers. We'll be going over this eventually, so I'm not focusing a lot on it right now. Following that, we looked at the properties of adaptive immunity, like antigenic specificity, diversity, immunologic memory, and self-non-self distinction as well, or self-non-self recognition as well. And we took up each of these topics in more detail subsequently also. Following this, Following this, we understood the key players of adaptive immunity, which are the B cells and the T cells. The two subtypes of T cells come in next, which are the T helper and T cytotoxic cells in this case. We understood the different surface antigens which are present, which now we know that either they are B cell receptors or T cell receptors. And in addition, we found some other surface antigens as well, which are extremely important. And we have understood how these co-receptors or co-stimulatory reactions or interactions need to be present in order to complete any particular reaction between the T cell or between the B cell and T cell, or whatever sort of immune reaction we are looking at. And we had understood how they are actually distinguished, which are like these antigens are actually classified or are named depending upon the structure that they possess, the sequence that they possess, and they are assigned into different CD classes, which are cluster of differentiation. So they are either CD4, CD8, or several other types of molecules which are present on the surface. In particular, because T helper cells always carry the CD4 antigen, they are known as CD4 positive T cells, whereas TC cells are CD8 positive T cells. So that was the co-stimulatory participant in any immune reaction. Apart from that, we understood that there are certain cell membrane molecules which are also very important contributors. Like we just saw, of course, the membrane-bound B cell receptors, T cell receptors, and in addition to other molecules, which again we have discussed in a separate week's topics as well, which are the MHC molecules or the major histocompatibility complex molecules. All of which altogether at the end of the day are serving the same purpose, that anything which is foreign to the body is recognized, properly recognized, and in response there is an appropriate reaction which helps in removal of that pathogen. In addition, that removal should be accompanied by a memory as well, so that in case of future infection, response can be elicited to quickly clear it out from the system. So that is the overall idea, which is sustained by adaptive immunity. Next, we try to understand the distinctions between humoral and cell-mediated immunity. Humoral, the name comes from humors, which means the liquid part of the body, and that actually involved the antibodies because they were found to be circulating within the blood within patients. So that is how they were initially identified. So that antibody-mediated immunity is known as humoral immunity, whereas all the T cell mediated immunity falls under the branch of CMI or cell mediated immunity. Next, we understood the difference between these as well, how cell mediated response and humoral response actually distinguish from each other. But effectively, the downstream response that is found to be the consequence of this recognition is also different. For example, in case of cell-mediated immunity, there is cell death via cytotoxicity, whereas for humoral response, there is the binding between antigen and antibody, and then its clearance can actually occur in several different types of mechanisms. In case that T cell is able to communicate, that B cell is able to communicate with the T cell branch of immunity, then there can be cytotoxicity in that case as well. That is, it calls for or it connects the humoral and cell mediated branches of immunity and facilitates the removal of the pathogen from the system. But there are other alternative mechanisms for this as well. So that's also something we have looked at. Next came the idea that it's not as simple as the fact that a pathogen enters, a B cell or a T cell recognizes that pathogen, and then it clears it off. That's not enough. There are a lot of sub-regulatory steps which come in. And one of the key steps that are very crucial in order to facilitate clearance of a pathogen. Recognition is one thing, but the response or the clearance of that pathogen will actually involve a lot of amplification. What do we mean by that? It means that if one particular, say for example, um, let's think of a mall where there is a particular, a thief that has come in, and one security guard recognizes that thief. That security guard is not enough. You need all the guards to be able to recognize that, or you need more of that same security guard. Of course, that's not humanly possible, but just conceptually speaking, expansion or amplification becomes a very crucial step. What also needs to be kept in mind is that just amplification is not necessary. Amplification, while maintaining specificity, is very crucial. So the specificity for that particular antigen should be maintained. That means you do not recognize any innocent passenger as a thief. You make sure that only the thief is getting recognized as the thief, while the number of people who are able to catch that person is increasing in number. So that's exactly what happens during clonal selection and clonal expansion. Selection means you are from a sea of B cells, only those B cells which are able to recognize that particular pathogen are preferentially selected. And once selected, they are expanded in number so that instead of one person, there is an army which is able to locate and remove that particular pathogen from the system. So that is what happens as part of clonal selection. Now, after recognition is facilitated, next comes antigen processing. So that processing is also something we have discussed extensively, and it differs based on what type of pathogen it is. If it is intracellular in nature or not, depending on that, the response will be different in each case. So either that pathogen has to be taken in or that pathogen is itself intracellular in nature. Depending on that, the way in which it will be presented on the cell surface will also be different, and accordingly, the way in which it will be cleared from system will also vary. Next, finally, we had talked about the fact that the immune system can often not work properly. Again, we have looked at this extensively later on, where we saw that immune dysfunction can lead to a large number of infections, infectious diseases, and in a significant number of cases can often even lead to cancer. But apart from that, some other common diseases include allergy, which we have just now discussed as part of the hypersensitive response, asthma. We talked about graft rejection, graft versus host disease, autoimmune disorders, and immunodeficiency as well. So with this, we had concluded the discussion for week 1. As part of week 2, again, please interrupt me at any point of time. I'm going a little fast because like there are 12 weeks to cover. It's going to take a little while. So that's why I'm going a little fast. If not clear, please interrupt me. As part of week 2, we had ventured into what exactly is innate immunity in more detail. So yeah, so we had talked about the different aspects or attributes of innate immunity here. The first was PAMP triggered immunity or pathogen associated molecular patterns. Effector triggered immunity, a recognition of the absence of self, that if self is not present, then innate immunity comes in. If there is altered self, that is, if a self cell or a host cell is undergoing some sort of transformation because of, say, for example, if it is cancerous in nature, if there has been a viral infection within the cell and it starts behaving in an abnormal manner, then in such a case, innate immunity can be very relevant. Then, if a cell is damaged, then its recognition and under default conditions, surveillance to make sure that a cell is healthy and it is not carrying any unwanted proteins, or it is not behaving in an abnormal manner, basically. So those are all the different aspects that are monitored and that are a part of the surveillance mechanism of innate immune cells. Following this, we understood the different properties. We looked at the participants of each of these subtypes. For the innate immune system, there are neutrophils, basophils, eosinophils, monocytes, macrophages, mast cells, and NK cells. Again, NK cells are very crucial with respect to the job that they do in context of cancer. We looked at the adaptive branch of the immune system as well, whereby there are T cells and B cells which are playing the major role there, and how dendritic cells are bridging the two. Again, this is a question I think that was part of the assignment as well. We had discussed in part of some sample questions also that how are dendritic cells bridging the two? Like, first of all, which cells bridge the innate and adaptive branch? What is their job? How are they actually facilitating this? So all of that is something we have studied extensively. And what I also want you to connect this with is the fact that dendritic cells are extremely efficient, or rather the most efficient professional antigen presenting cells as well, which actually is one of the key reasons why they are able to connect these two branches of immunity also, and therefore they are very indispensable to the system. Sorry, I'm getting sore throat. So that was part of the innate immune system. The modulators or the mediators of innate immunity majorly involved two subclasses. Not two subclasses, a major subclass was pattern recognition receptor. So again, the distinguishing factor between innate and adaptive immunity was the fact that innate immunity very extensively relied on the presence of patterns on the surface of their pathogens. What do we mean by patterns? Either it could be the presence of lipotoxins, DNA, RNA, or some endogenous danger signals, beta-glucans, double-stranded RNAs, etc. Depending upon what sort of pattern the immune system was able to recognize, in that case, that pattern acted as the ligand. The response to that was elicited by receptors which are present on our immune cells and or present in our system. And the source of the ligand could either be bacterial, viral, parasitic, an altered host cell, or in some cases fungi, in some cases RNA viruses, etc. And in response to that, an innate immune response was elicited. On the right, you can just find some abbreviations for certain common terms. So I'm not actually going through that. Following which we understood in more depth how toll-like receptors or TLRs work. They are indispensable when we talk about the innate immunity. We looked at the different subclasses. Apart from TLRs, there are also C-type lectin receptors. You have NOD-like receptors, all of which manifest these cellular responses, either phagocytosis, cell death by autophagy, apoptosis, or inflammasome formation. So we actually looked at all of this as well. Next, we came to the cells that actually participate in this context, and we understood the role of macrophages, neutrophils, and monocytes, and how they exactly respond as part of the innate immune system as well. Majorly, a key role of the NK cells were understood in this context, how they have different roles to play depending upon what type of stimulus is added in is present or is added in a particular condition. Say, for example, if there are certain interferons or certain chemokines which are present, then they can lead to some very specific responses, and therefore overall have a very huge role to play, at least in terms of innate immunity. So we understood those as well. Next, here we had looked at the difference between inflammation and swelling, particularly because inflammation was very relevant to innate immunity. We understood edema, erythema, heat, pain, and loss of function as the key attributes of localized inflammatory response. And just a few slides ago, if you remember, we saw the role of edema and erythema, particularly in vasculitis as well. Again, if you don't already are not familiar already with it, then itis, the term, any disease condition or any condition which ends in itis refers to an inflammatory condition. So there has been some sort of a allergic or an inflammatory response, and therefore that condition has itis at the end of it. So you have conjunctivitis, again you have vasculitis, you have meningitis. They are all inflammatory conditions. So that's an easy way of recognizing a subtype of diseases. Next, we have the process of inflammation. Again, we have discussed this extensively over multiple weeks, actually, how in case there is a tissue damage, in response to that, how you can find vasoactive mediators and chemotactic factors being released. And as a response to that, there is the influx of exudates and cells from the capillaries, which are permeable in nature, into the surrounding tissue in response to the injury which has happened. And as a response, phagocytes will actually move towards that site of injury and will clear out or exudate or destroy whatever pathogen had come in. So that was exactly their job. And we had discussed all the sub-steps that are part of it, how there has to be margination, transendothelial migration, there has to be CAM, ICAM recognition. Please try and remember these interactions as well. If you remember, it was also part of the assignment questions that between the cell that is migrating and the surrounding capillary environment, what are the different types of interactions that actually occur. So please make sure you remember and you like brush up on this very clearly, because we have discussed everything. All the material is available to you. You just need to go through it very quickly once. Next, these were the common principles of innate immune effector mechanisms, like we already discussed. There can either be antimicrobial peptides, antivirulins, antitoxin effectors, protease inhibitors. We have lysosome-mediated destruction as well, cell death, and other immune effectors, all of which can execute the job of the innate immune system. Next, we talked about the organs. We had discussed the thymus, how this was the cross-section schematic representation of the cross-section of the thymus, the different cell types which are present, each of their functions. Just to quickly remind you, make sure you brush up on what the nurse cell actually does in the thymic cortex, what is its job, and why is it almost always indispensable. So make sure you read up on these subtle differences as well. They might be very important. Then this was the cross section of a lymph node, which again is also part of the immune organs and immune system organs. And these are its different components. We have extensively gone over all the sub-steps that happen here. In particularly, make sure you brush up on what germinal centers do, why they are relevant, what exactly is happening inside the thymus, inside the lymph node. All of that we have discussed, and we'll be coming to some of that in the subsequent slides. Sorry, just a second. Then we had briefly discussed how B cell development proceeds. There are two major phases, antigen independent and antigen dependent phase. We got into a lot more detail about this in the subsequent slides. But again, what I would like to reiterate are the sites or the locations basically within the body where each of the developmental stages actually proceed. So you can see how it begins with the bone marrow, and then the peripheral lymphoid organ also gets involved. And each of the what is the role of each of the organs, that also you should be very clear about it. That while in the bone marrow, only the differentiation, partial differentiation, or rather more accurately, you can just say gene segment rearrangement step. It occurs within the bone marrow. All the development, it occurs within the peripheral lymphoid organ, depending upon what type of antigen this particular cell is able to see. When I say see, I mean what it is able to interact with or what it carries specificity for. So depending on that, each of these steps are spatio-temporally separated. We got into more details about the way in which B cell develops eventually, and what are all the different sub-steps that are also present, the different zones which are also available that involves the light zone and the dark zone, and how antigen-stimulated B cells actually undergo all of this differentiation to make the possibility of clonal selection and clonal expansion at all possible. And also what you should definitely revise from here is the sequence of events. I don't think I have it on me right now, but you need to make sure that there was one particular question, I think, in the assignment this year, where each of the sub events that occur, they have to be arranged sequentially. So you must know the order in which each of these events are occurring. So please, you know, make sure you are well versed with that. So yeah, so then we understood how T cells actually develop in mice. So it starts off, of course, with the stem cell, which is the hematopoietic stem cell, and the associated surface markers are also very important. How it undergoes migration, it undergoes extensive genetic rearrangement, and while it's very energy, when I say energy, I mean ATP or resource consuming within the cell. It's also a very, very crucial part of any organism survival, and therefore it's an affordable risk for the cell, energetically speaking. So, sorry. So these cells will actually undergo locus rearrangement or genetic rearrangement, after which they will migrate through the blood into the peripheral tissue at the site of antigen entry. So with that, we had come to an end of the discussion for week 2, following which in week 3, we understood antibody structure in a lot of detail. Again, what you definitely need to remember is the antibody structure extensively. It's not very difficult, but it's very, very relevant. So make sure you are very clear on basic questions like, say, for example, the linkage or the bond that is present that holds the entire antibody structure together. If in case we can see there is removal of disulfide bonds, then what will happen to that protein? What would happen? Say, for example, when I mentioned the fact that you will have a section in your exams where there'll be a slightly more application based or more detailed question or concept driven, high concept driven questions, then in such case, what you know needs to be applied. So for example, you might be asked about the fact that if you have one X antibody and then you digest it using papain or you digest it using pepsin, then what is like if we digest it and then we try to visualize it, separate and visualize it, then what are the type of bands we will get in such an STS page? So such questions can only be answered if you are conceptually very sound. So make sure you don't have any queries about that. Um, so what you see here is the difference between affinity and avidity. Again, a very important, very relevant difference. So affinity refers to the strength of one single antibody or one single antibody with its antigen that it is interacting with, and each IgG antigen binding site. But okay, each IgG antigen binding site typically has a very high affinity for its target. And on the other hand, you can see avidity, where the strength of all the interactions are actually combined, and therefore IgM is most important, most relevant in this context. So avidity in such cases would be high. So then these are all the different sub-steps that are part of the antibody developmental process, the different structure or the structural differences between different um, groups of antibodies, IgG, D, E, A, and M. So again, if you don't remember, nomenclature is based on alpha, beta, gamma, delta, epsilon. So that's how antibodies were actually named. And also, you need to make sure that you can see the difference there between each of the antibody subtypes, either in terms of the number of disulfide bonds that are present, what is the number of, say, for example, when we talk about IgM, the fact that there are 5 antibodies that are all together forming that pentameric structure, such key difference points should be clear to you if it comes to you in a question directly. Say, for example, you're asked about a peptide that is present in one particular antibody. Then in that case, IgA certainly has the secretory peptide that is attached to it, and that would be a key distinguishing factor in such a context. Then we had a lot of discussion about Tonegawa's model, or how Tonegawa had actually made the discovery about these cells, not these cells, but rather the entire process of gene segment rearrangement. He had made that extensive discovery about gene segment rearrangement that occurs within these cells, and the experimental evidence for the same. What is the expected blot that comes out as a result of this experiment? So something like that can also come as a question for you. So you can look at that. Then we had gone over the process and what are the different types of gene segments that are present, each of the heavy chain and light chain elements, how many segments are present, how that exact process takes place, what are the enzymes involved. All of that was discussed. So I'm not going over it because we have extensively discussed it previously as well. This you can see the kappa light chain DNA, the heavy chain DNA, the key differences between these substeps, how there is VJ joining and DJ joining as the first step of each subtype, followed by VDJ joining, and then there is difference in polyadenylation and RNA splicing, which leads to different types of chains being synthesized. So depending upon where splicing is occurring and where poly A tail is getting attached, you can have different types of heavy chains, basically. So all of that was also looked at. The sources of antibody diversity, we have studied how there can be either multiple germline gene segments, combinatorial joining, junctional flexibility, P region and N region nucleotide addition, somatic hypermutation, and then their combinatorial association, antibody diversity. Following which we had understood the process of signal sequence and how their orientation, recognition signal sequence, or RSS basically, and how their orientation, whether or not they're facing each other or they're facing away from each other, you can find different generation of different types of antibodies. So that actually leads to differences in terms of either deletional or inversional joining, leading to different consequent types of DNA, which can therefore generate more and more antibody diversity. Then we had understood junctional flexibility as well, that depending upon where two fragments or two chunks of DNA are able to associate with each other, there can be flexibility there as well, which can generate or which can add more to the existing antibody diversity. Following this, we made an approximate calculation of the number of antibody subtypes that can actually be generated if we take into account all of these contributors. Then there was also another key contributor, which is in terms of P nucleotide and N nucleotide addition, and how in each case, how exactly is this facilitated that a nucleotide is getting added and what is its role is something we have already discussed. You can go back to the video lectures if you need it. Then, yeah, so that was week 3. Then in week 4, this was just a quick revision of antibodies and B cells. Then we understood the different subtypes in terms of secreted and membrane-bound antibodies, how that difference is actually brought about in terms of different polyadenylation sites, either PA1, PA2, PA3, or PA4. Each of these has the potential to generate either IgM or IgD subtypes of antibodies. Then we have RNA transcripts for either the secreted subtype or the membrane-bound subtype. And when membrane-bound, it has an entire additional segment in terms of either the membrane-bound or the inside or cytoplasmic chunks. Or on the other hand, it can be completely secretory in nature when it will be a smaller protein. So we understood these some key differences between secreted and membrane-bound subtypes of antibodies. We understood different types of post-translational modifications which occur in that case, different types of alternative splicing events that actually contribute to the same. It's not always just the same, mutual excision of exons that is always necessary. It's not that it is always necessary, but there can be other conditions as well. Either, for example, there can be constitutive splicing. There can be mutually exclusive exons, cassette alternative exons, alternative 3' splice sites, 5' splice sites, and intron retention. Following this, sorry. Following this, we understood the differences between isotype, allotype, and idiotype, the key difference points between them, what are the definitions, how you can actually distinguish them. So you can go over it quickly, but basically, if we just briefly explain it, then isotype will actually be defined by the constant region of the heavy chain. So for example, this is the IgG1 from mouse, and this is IgM from mouse. So in that case, it's heavy chain constant region and the surface antigens on it are exactly what matters. The binding specificity of all these antibody subtypes do not really change. And then comes allotype. In allotype, you have antigenic determinants that are specified by allelic forms of these genes. So allotypes are an immunoglobulin variation found among classes of antibodies. On the third type, if you take the example for it, it is strain A and strain B IgG1. So they are allotypes of each others. Third, you have so in this case, what you can distinguish it from is the isotype and the idiotype. In case of idiotype, the variation comes in the variable region. So that is a key distinction in that case. So just to quickly summarize the difference between them. In the first case, they are isotypes of each other, which means that variable region specificity is the same, and difference is found in the constant region. In the second case, we have allotypes. So in that case, also you can see that antigen specificity is unaltered. What is changing is the constant region, heavy chain constant region antigenic determinants. And in the third case, you have a difference in the antigen binding region or the variable region, which are known as the idiotopes. This was a comparison between each of the different antibody subtypes, and a note that I had made here was that the IgG is the only antibody subclass that significantly crosses the plasma membrane or not the plasma membrane, the placental barrier. And not all subclasses among IgGs can actually undergo transplacental migration with equal efficiency. There is a differential ability that distinguishes them. So in this case, say for example, you can see IgG 1, 2, 3, and 4, and you can see how their propensity of crossing the placenta can actually vary among them. Then you have isotype switching, and we saw it extensively before, where we understood how, depending upon the type of condition that the antibody needs to cater to, the most efficient isotype is chosen, and that is facilitated by isotype switching, which is mediated by cytokines. Then we had a brief introduction to MHCs, major histocompatibility complex, and the different subtypes under that, MHC class 1, 2, and 3, and how actually their gene products or uh, um, the different sub-classifications are actually understood. Then we understood the key difference between type 1 and type 2 MHCs. Structurally and of course functionally, both of these were taken into consideration. The gene segment rearrangement, that brief, not even a gene segment rearrangement, but rather just the um, schematic representation of what exactly happens with respect to MHCs class 1 or class 2 is what we saw here. Then we compared the working mechanism of or the ability of peptide binding by the MHCs and what exactly um, what exactly distinguishes them from each other and what is the nature of these molecules as well. On the right, you can see examples of anchor residues, which are shown in blue. For example, here it is tyrosine, here it is glycine and glycine actually, which plays a major role in this case. So that along with proline is a very unique motif to be present and therefore could be relevant. Then in week 5, we looked into MHCs in more detail. We understood the different subcomponents, the arrangement of the of the MHC genes, basically in the role or the activity of these MHC genes that was also studied and how they are all localized along the human chromosome number 6 and what exactly is their job there. Then allelic forms of MHC genes gave birth to the terminology known as haplotypes. Their inheritance was studied, the phenomenon of linkage disequilibrium and how it actually makes a difference. That was also studied. We understood experimentally how MHC inheritance actually occurs with skin transplantation experiments. We had, yeah, here's what we had actually seen and how acceptance and rejection of skin grafts and the consequent coat color that sets in as a response to that was also studied. And this was the schematic representation of the way in which HLA haplotypes are actually inherited in a typical, under normal conditions in a particular family. That was also understood. Then the different attributes of it, how it is polymorphic in nature, how inheritance can depend upon a number of factors, that was also studied, how amino acid sequence variability is relevant to generate diversity within a population in the MHCs, to generate diversity in the MHCs within a population was also extensively studied. Then we made a comparison between the efficiency of all of these um, uh, uh, not efficiency, the role of these components with respect to the type of pathogen that is coming in. So depending on whether it is a cytosolic pathogen, an intravascular pathogen, or it's an extracellular pathogen, and whether toxins are involved or not, the type of cell and the type of response can actually vary. So it is either you can see an effector CD8 plus T cell, effector CD4 plus T cell, and the type of response in each case can be distinguishing or distinct from each other. Then here we understood antigen presentation in more detail, the role of tapesin, TAP-associated protein, etc. All of this was extensively studied, and particularly the role of the Golgi and the clearance that was studied. So here we had understood how tapesin works, TAP1 and TAP2 are involved. And one condition that is often associated with this disease is necrotizing granulomatous lesions which were found in the TAP deficiency syndrome. So while it might seem like nothing, the presence of tapesin is extremely crucial for proper development and basically for responding to toxins in the environment. Then we understood the antigen presentation in more detail, how class 1 MHC is getting localized and followed by the generation of antigenic peptides in the exogenous um, processing pathway as well. So again, this was a topic we had discussed previously also. Then the role of the invariant chain and the clip protease and how it releases or rather removes the tail end and releases the clip protein for its own activity and assists in the overall assembly of class 2 MHC molecules. On the right, you see a schematic which actually compares these two pathways, the endogenous and the exogenous pathways, respectively taken care of by class 1 and class 2 MHCs, and the different substeps that are included in them is also being mentioned. Then, as part of week 6, we had looked at immunodiffusion techniques. We had deviated a little bit and started going towards the technical part associated with immunology, the formation of precipitants. I think I'll go over this a little fast because it's more not really very concept based. It's just experiments back to back. We had understood the differences between SRID and ODD. Double immunodiffusion and radial immunodiffusion were taken as into consideration. The SRID or single radial immunodiffusion condition versus the double diffusion tests as well, and how you could correlate the area of the precipitin ring. And you can use it to determine or estimate the unknown concentration of the antigen sample. This was octolonase double diffusion or ODD, and how depending upon whether the two antigens you are studying are identical, partially identical, or non-identical, that recognition you can do based on the type of precipitin lines that you can see. Then we had also studied ELISA. So enzyme-linked immunosorbent assay was extensively studied. And then the basic processes and how it is similar and different from Western blotting, all of that we have already addressed. In case it is something that you don't have on your mind right now, you can read up on this topic, like we have discussed it before as well. The different subtypes of ELISA were also taken up, including indirect, direct ELISA, sandwich ELISA, etc., comparative ELISA, all of that. Second. This was Western blotting. We had looked over each of the different steps. I had shown you how this gel is cast, placed within the buffer tank, samples are loaded into it. Then how you can facilitate transfer of the transfer of the protein samples from the gel into or onto a membrane. That membrane acts as a solid support, and then that membrane can be probed to check for what proteins are present. So this was the transfer unit and transfer assembly for moving proteins from the gel onto the membrane. Then we had looked at immunofluorescence as a technique and the different compounds that participate along with it: fluorescein, rhodamine, DAPI, excess red, etc. This was a fluorescence assisted cells, like it's a cell sorter basically, or a system which allows you to segregate your cells based on a particular criteria. Let's put it that way. So we had understood how FACS works and how it is often used in conjunction with a flow cytometer basically. But yeah, FACS actually allows you to collect the sorted cells and use it for your experiment, whereas flow cytometry will not actually be able to do so. Then as part of week 9, we looked at T cell development. Again, the same steps which are involved there. We then looked over the thymocyte signals and how they are relevant. Then the different substeps starting off from a double positive stage, from the enzymes that are involved that are actually facilitating the process, including RAG1 and RAG2. All of this are relevant here. Then thymocytes, they undergo two selection processes. We had understood those about positive selection and negative selection as well, and how both of them are actually necessary to complete this. I'm not going over this because, again, we'll run short of time if we go into so much detail about each of these steps. But basically, there is extensive selection, which is necessary in order to complete positive and negative selection of T cells within the entire system. Here you can just don't know, some issues there. Okay, anyway, here we had looked at the T cell mediated signaling. What is the downstream signaling that is associated with it? The signal transduction pathways, including phospholipase C gamma, also came in this regard. So that actually these were the downstream signaling, which happens much later. So all of that also is something we had looked at. Then we understood the co-stimulatory signals, which have a very important role to play here as well. So that starts off with the B7-CD28 reaction, how in some cases IL-2, IL-2R interaction is very important, which leads to generation of memory and effector B cells also as well. This is a comparison between each of these different subtypes. When I say subtype, I mean each of the different key players in context of antigen presenting cells. So this also was something we looked at. Coming to week 8, we focused on B cell development. Again, this is a familiar figure. Then we looked at all of the different substeps which have a role to play here. This is much similar to that of T cell development as well, the different interactions which are necessary in order to facilitate B cell maturation. That could be very relevant. So you should definitely make sure your concepts regarding this topic are like in absolute confidence. Then this is B cell maturation. So the different substeps, how there is H or heavy chain and light chain present. And then we have RAG1, RAG2, TDT, membrane IgG, transcription factors, and surface markers as each of these steps progress. Again, this is a relevant figure, so make sure you remember the subsequent steps so that that is also conceptually clear. Then we looked at the experimental evidence for this as well, how there is also a possibility that there are three possibilities. Either mature B cells can be like as part of clonal direct deletion, any B cells which are self-reactive can be cleared off, or there can be light chain editing or in order to allow it to persist, or there can be energy as well. Then we have T cell antigens. I think there's some issue with my. Just give me a second. It's working. Okay, we went over the different types of T cell antigens as well, following which is the issue, but yeah, let's see. Following which we understood B cell mediated signaling in more detail, how there is signal transduction downstream of it, and how that is different from what we have already studied, what are the signal transduction pathways. Then thymus-dependent T cell activation, thymus-independent activation is already familiar for us. We understood the substeps of B cell proliferation, how cytokines have a different role can have different roles to play there. Then as part of week 9, we ventured into the complement system. The cascade, this was the discovery of the complement system, how there can be different downstream responses. Either there can be cell lysis, there can be opsonization, inflammatory response, and clearance of the immune complex so as to remove the pathogen from the system. How there are 3 pathways that are merging in order to allow the complement system to function. How the major amplification step is the generation and the functioning of the C3 convertase, the classical alternative and the lectin pathway or the mannose binding lectin pathways are all merging at this final step. I'll not waste time over this. But yeah, these are the substeps of the classical pathways. You can go over it. These were the alternative pathway inducers or initiators, how certain microbial pathogens and non-pathogens, both of them participated in this substep. The regulators of the complement system, especially the shorter segments that are generated as the result of cleavage in each subsequent step. Then the regulation of complement system at different levels by different inhibitors at different substeps, either before convertase assembly or after it, or at the step where membrane attack complex formation will occur, and what are the different mediators of this regulation as well. So this showed the biological effects of the complement system, what exactly it does downstream. And here you can see some scanning electron micrographs showing you the difference between these activities as well. Then in week 10, we extensively looked at what are cytokines, how they are communicators within the cell, how they can actually have different sort of downstream effects. Either they can be pleiotropic in nature, redundant, synergistic, antagonistic, or just cascade inducers. How combination and combinatorial or individualistic presence of certain cytokines can have certain types of downstream reactions. Then the functional groups which are relevant in this case, different cytokine receptor families. Particularly, we focused on the class 1 family in more details, example being the GM-CSF, IL-6, etc. Among these, we also looked at the type of response which can be generated depending upon what combination of receptors is present. So you can have low affinity or high affinity receptors depending upon whether it is one subunit or two, and accordingly the reaction may or may not vary. Yeah, so that's what you exactly see here, that low affinity IL-2R only has alpha subunit, intermediate affinity has the beta and gamma, and when all three are present, it has the highest affinity. And actually the IL-2 receptor is very, very important, and it's one of the prime examples used in this context. We discussed SCID as well, congenital X-linked severe combined immune deficiency as well, mapping to the X chromosome. So this was just one example which we saw in more detail, and this showed more elaboration of the signal transduction pathway and cytokines and how they are relevant to cell-mediated immunity. Then the therapeutic use in a commercial manner or otherwise in clinical context, whatever is necessary, and what are the diseases that they are usually concerned with. We had looked at that as well. We understood hypersensitivity in more detail. We looked at type 2 and type 3 today, but of course type 1 and type 4 were already covered that day. This was type 1, the principal mediators of type 1. This was an example of skin testing that is done to screen for potential allergens. Then you have type 4 hypersensitivity or delayed type hypersensitivity as well, and the type of pathogens that are relevant in that regard. Yeah, so this was type 4. For week 11, we looked at tolerance and autoimmunity, the type of response it is, whether it is central or it is peripheral in nature, the different definitions that distinguish immunogens and tolerogens from each other, the factors that aid tolerance and that mediate tolerance within the system were also very important. This we understood central tolerance in more detail and how gene rearrangement is extremely important there, followed by peripheral tolerance, where the cell can have three potential fates: apoptosis, energy, or regulation, and for self antigens that may be present in peripheral tissues. Then this was C.C. Goodnough's experiment for the concept of central tolerance to at all be taken into consideration. Um, and yeah, these are the potential fates, like I just discussed. The existence of Treg cells and how they exactly end up doing their job. How do they actually maintain the regulation? All of that was extensively looked at. Then we looked at some examples of autoimmune diseases, either organ specific or systemic in nature. The inducer or the self antigen which induces that immune response was studied with very concrete examples of, say, for example, organ specific diseases like Hashimoto's thyroiditis, Graves' disease, or which involved stimulating autoantibodies. On the other hand, there was another example of myasthenia gravis, whereby there was a blocking nature of these autoantibodies. Then we have systemic diseases, example being systemic lupus erythematosus as the prime example for this condition as well. This was an example of the same. Finally, the release of sequestered antigens and how they could potentially induce autoimmune diseases is, is an is a key concept, because you need to understand how sequestered antigens are not seen by the immune system, and as a result, in case of any trauma or in case of any unexpected condition, their exposure would actually would carry very high potential for becoming an autoimmune disorder. Then the concept of molecular mimicry, and finally the role of autoimmune regulator was studied. In the last week, we understood vaccines and their role, how Edward Jenner and Louis Pasteur had a role to play there. We looked at early vaccination studies, the experiments that helped the same, the role of immunization, and the basic difference from vaccination, the two methods or two subtypes being active and passive immunization, different types of vaccines, whether live attenuated, inactivated, toxoid, subunit, conjugate, DNA, or recombinant vector vaccine. Then the types of immunization, natural active or artificial active, and their examples should be very familiar to you by now. Then we looked at the different dosage that is usually recommended by the Indian Academy of Pediatrics. So that gives us a real life idea of how drugs need to be administered. Then we have vaccines. The concept under vaccines for booster doses, which are basically repeated inoculations with the same pathogen, and how that altogether cumulatively leads to a much stronger immune response, and how it is relevant in context of polio. Not an immune response, but rather what I meant was that an immune response needs to be elicited for all three subtypes, which is usually difficult because of the nature of the pathogen, but how that would be difficult to actually manifest because viruses can undergo mutation and therefore they're difficult to tackle. Then whole organism vaccines, their methods of attenuation. Again, this we just discussed last week, so I'm not going into unnecessary detail about it. The way in which the BCG vaccine is made, the result, like the reason why it is named as BCG, then whole organism inactivated or killed vaccines and their examples, the distinction between the oral polio vaccine, Salk and Sabin types, that should be very clear to you. Then the subunit vaccines and their examples, how the diphtheria toxin is relevant in that case. I discussed the concept of conjugate vaccines and majorly looked at one example for the same, which is under consideration and can be a very useful idea in this context. So with this, we have actually come a full circle for all the topics that we wanted to discuss for today. If you have any doubts, this is the time. Please go ahead and bring it up, and then we can conclude the session for today and, of course, the course as well. Okay, then if not, thank you so much for joining this course. I hope it was relevant to you and helpful for you. Again, this lecture as well will be uploaded, but your exams are pretty soon, so I don't whatever help you can get out of it, please do. If there are any doubts, you can write down in the discussion forum, or you can also mail anybody from the team and they can get back to us. Apart from that, for reference, like I already mentioned, you can go to Kubi Immunology for slides related to what I have discussed, or any immunology book for that matter, whatever is accessible to you. Go over the slide notes, go over Professor Shudeep and Professor Ganguly's lectures, just quickly go through the transcripts at least so that you can brush up on what they have discussed, because they have given a lot more information than what is just normally present in books. So that can also be very important, and prepare well. And all the best, and thank you so much for joining. We will be ending the session and discussion for this course today. So thank you so much. Have a good day and all the best for your exam.